Hi, I am Parijat Senapati. I am a staff scientist at City of Hope. I work on cancer research. Only 2% of our DNA is genes, and the rest 98% is basically a black box. All the initial cancer research was directed towards studying genes, but nobody knew about this rest 98%, and it was thought as a black box or junk DNA. But we know now that the rest of the DNA is not junk. They also have something, and what they have is something called as transposable elements. So transposable elements are basically, simply put, initially when they were discovered, they were called as jumping genes. Basically, these DNA elements come through viral infection. So a virus like a retrovirus, basically when it infects cells, it's an RNA virus. It can convert its RNA, which is a genome, to DNA, and that DNA can attach itself or integrate into our DNA, and they can copy themselves and jump to another place. So as you can imagine, that this is not good. There are systems in place to check them, but in cancer, these same systems are lost, and one of the functions of these tumor suppressors is also to silence these jumping genes. So I'm hoping to show that by studying the DNA sequence and the RNA sequence of the tumors, I can compare and find out whether these junk elements or these transposable elements or jumping genes are being expressed. And since these will be tumor specific, they will be expressed only in the tumor cells, because these are the only ones that have lost this suppression. This would help us identify or target the tumor specifically. The most direct clinical significance of my research will be the discovery of what is called as cancer-associated antigens. So basically, these are proteins that are expressed only on the cancer cell surfaces, and this will help other scientists increase the target search. So they will be able to design immunotherapeutics or CAR T cell therapy, which can target these cells specifically, and they will target only the pancreatic nets and not other genes that we already know of from other cancers. Scientific research is very slow and very expensive, without much rewards for us scientists. So it's the faith that the donors put on us is encouraging, and it also again helps fund our research, because a lot of scientists have ideas, but then there should be funds to execute them, and if not for the donors and the funding from NETRF, that was not possible. So I'm very grateful, and thank you to all the donors who support scientific research.